I want to welcome everybody and thank you for joining us. I'm Todd Wilkinson, the 2023 NCBA president. I'm a cow-calf backgrounder and feedlot owner from the little town on the prairie in DeSmet, South Dakota. Tonight's topics are certainly valuable and going to provide some great insight. I can tell you South Dakota is again bracing for another winter storm in the next 24 hours. It seems like it's the winter that's never ending. You know, we got an excellent panel tonight, and as a participant, your line will be muted, but feel free to type in questions in the question box on your screen during the webinar. And at the end of the presentations, we will get to as many of those questions as time may allow. If you have trouble with your technology or if you're joining us only for the audio, the webinar is being recorded and will be available for viewing in a few days at the ncba.org site. Just look for the producer tab on the website. On the webinar tonight, entitled New Vaccine Technologies, in addition to our Animal Health Toolbox, we'll feature Dr. Bruce Thompson. Dr. Thompson is a senior staff veterinarian in the Policy, Evaluation, and Licensing Unit of the USDA Center for Veterinary Biologics, CVB. He obtained his DVM from the University of Missouri and started his professional career working in a mixed animal practice for 7 years. He then went to Iowa State and received his PhD in anatomic pathology. Dr. Thompson is a pathologist at the USDA National Veterinary Service Laboratories was a pathologist prior to joining CVB in 2016. Next, we have Dr. Chase. Dr. Chase is a professor for the Department of Veterinary and Biomedical Services, South Dakota State University. He received his Doctor of Veterinary Medicine from Iowa State University and his MS and PhD from the University of Wisconsin-Madison. He was an associate at the Viborg Veterinary Clinic in Viborg, South Dakota for 5 years and continued his active involvement in the clinical veterinary medicine for the next 25 years. Dr. Chase's research has been directed at understanding the initial events of viral infections to develop better methods for preventing virus infections in animals. His clinical experience is with dairy, cow-calf, feedlot, and hog herd health. Dr. Chase served as president of the American College of Veterinary Microbiologists until 2018. Finally, we're joined by Dr. Amelia Willems. Dr. Willems is the Michael and Mary Creek Hall Davis Endowed Professor of Beef Cattle Health and Reproduction at the College of Veterinary Medicine at Mississippi State University. Dr. Willems received her veterinary degree in 1988 from Purdue University. She then completed an internship at Kansas State University, a residency at the University of Saskatchewan, and a PhD at the University of California at Davis, where she studied the immune response of cattle to BRSV vaccination. Dr. Willems is a board-certified veterinary internist and immunologist. At Mississippi State University, Dr. Willems works in the Food Animal Teaching Hospital, and she conducts research on BRD prevention, antimicrobial resistance in BRD, and the vaccination in cattle and calves. At this point, I'd like to turn it over to Dr. Thompson. And Dr. Thompson, you have the floor.
Thank you very much. Thanks for this opportunity to talk about new vaccine technology and how CVB evaluates vaccines this evening. The USDA CVB licenses animal vaccines, and my job is to evaluate new vaccines that are proposed for licensure. We're based in Ames, Iowa, and the picture in the first two slides is our facilities at the National Centers for Animal Health. There are many ways to approach this broad topic of new animal vaccines, and there are many steps in the licensing process. So in this short amount of time, I'm going to limit my discussions to safety evaluations and won't be discussing all the other many requirements. And after I paint the big picture on vaccine safety, then Dr. Chase and Willems are going to follow up and fill in the finer details on the different types of vaccines and how they interact with the immune system. And just now I briefly saw their presentations and they will be worth listening to. So I'm going to try and shorten my presentation so we can get on to the good stuff that they're going to talk about. So here's our mission. And this law from 1913 is why CVB exists, and it guides our actions. The need for pure, safe, potent, and effective animal vaccines was understood 100 years ago and is still relevant today. I'd say everybody that's on this webinar has a common interest in having the best vaccines available to prevent disease, whether you're a producer, a veterinarian, or a vaccine manufacturer. So what do we mean by pure, safe, potent, and effective? Pure, it means we know and understand everything that's in that bottle of vaccine and that it's not contaminated, because that would be a safety issue. Safe, we know we've done testing in animals and other types of testing to prove that it's safe both in the environment, and the environment would include people also. Potent, we know the bottle of vaccine has the right amount of active ingredients. And effective, which means the manufacturer has done studies to prove the label claim. So basically it does what it means, what the label says. And I'll call your attention to one more thing, the word available. We understand that we must create a licensing pathway that allows new, safe, and effective vaccines to get to the market. And that regulatory process is science-based and not excessive, because we all understand that there's new disease challenges as well as old diseases where we don't have good vaccines for, that create a really strong need for new and better vaccines. Sorry about that, I forgot to start my clock. So safety evaluations really start the first time we talk to the vaccine manufacturer. And it's kind of broken into two parts. There's the pre-license safety requirements and the post-licensure safety requirements. Pre-license is just that. Before it's licensed, we're going to require a wide variety of safety studies prior to licensure. Our laboratory is going to do testing on the vaccines and the components of the vaccines, and we're going to inspect the manufacturing facilities, make sure they're following their processes. Then after it's licensed, there are also requirements. We're monitoring the manufacturing facility, visiting the manufacturing facility, we're monitoring the sales, and most importantly, we're collecting adverse events. And again, science and product-specific data are going to direct our regulatory decisions. So when we do our initial evaluation of a proposed new vaccine, we're looking really at the big picture, that it's a scientifically sound vaccine and it makes sense. And then we're going to start characterizing the components of the vaccine used in manufacturing and how eventually the vaccine will actually be used. And in that, we do an initial hazard identification. And the picture in this slide admittedly are a little odd and kind of out of place, but I'm using them as examples to help explain the difference between a hazard and a risk. And in day-to-day language, we use these terms hazard and risk interchangeably, but in a scientific evaluation, they have distinctly different meanings. And it's important because we determine vaccine safety based on a risk assessment. So what does this mean? A hazard is something that has the potential to cause harm, while a risk is the likelihood of harm actually occurring. So in my first example, a lightning storm, we know from scientific data that if you get struck by lightning, that has a great potential to cause harm. So lightning would be considered a hazard. However, what is the risk or probability of harm occurring? We all know that if it's a sunny day outside, the risk or the likelihood of lightning striking us and causing us harm would be very low. Thus, lightning would be considered a hazard because it could cause harm, but the risk of lightning causing harm is very low on a sunny day. So our risk assessment would be that, yes, it's safe to go outside on a sunny day because the likelihood of harm occurring is very, very low. And now let me give you an example that's directly related to vaccines. Mineral oil has been used for decades as an adjuvant in vaccines to make them more effective. And when you add a little mineral oil to certain vaccines, it stimulates the immune response and again results in a better vaccine. We also know that mineral oil is the main ingredient in baby oil, and it's also a common laxative for both animals and people that many are familiar with, which is why I'm using it to illustrate my point. So if CVB would do a risk assessment to look to determine if mineral oil vaccine is a hazard, then what is the risk or probability of causing harm if it is a hazard? We have scientific data that shows mineral oil, when it's used as a laxative, whether it's in people or animals, and you follow the labeled directions and you drink a small amount of it, it's safe for both people and animals to consume. However, common sense would tell you if the animal person would drink very large amounts of mineral oil, it would clearly be unsafe and a hazard based on the volume. So again, in this example, just like the lightning, mineral oil would be considered a hazard because it has the potential to cause harm, but the risk of a very small amount of mineral oil when used correctly would be very low. So the risk assessment would be that mineral oil is safe. So what are some of the questions we ask in a risk assessment? Essentially, there's kind of three key areas that we look at. We look at the intended animal species that's going to get the vaccine, in this case, cattle. We look at public health safety, and then we also look at environmental safety. And when we're looking at the species that's going to get the vaccine, we're going to try and predict what the adverse events are based on the vaccine, what we know about similar vaccines. Then we're also going to look into whether there's some specific situations that might be a higher risk. Public health safety, we're going to look at the probability of human exposure, right? And since people are giving these vaccines to animals, there's always going to be the risk of human exposure. And then we're going to look at the possible outcomes of human exposure. Then we're going to do the same thing for the environment. And right, the environment is a very big place. It's other animals. So we're going to look at other animals, other things in the environment, see whether that vaccine could spread in the environment. Once we've done that initial risk assessment and answered those basic questions, then we actually start doing our studies. And the big one is the field safety study, where we put it out in the field and look for adverse reactions, adverse events. We do laboratory animal safety studies. We'll do overdose safety studies. If it's a live vaccine, we'll look at whether a vaccinated animal might be able to shed that live vaccine or not. If it's a killed vaccine, we'll do detailed inactivation studies to assure it's the virus or the bacteria in that vaccine is killed. If it's a food animal vaccine, we'll do injection site studies. And all food animal vaccines have a minimum of 21-day slaughter withdrawal. And certainly with some live vaccines, we also do testing in non-target animal species, meaning we'll see if there's a risk in another species, we'll put it in that other species to assure we're not taking unneeded risk. So during the licensing process, right, which typically takes a couple years or maybe even more than that, we're gaining all this information and we're putting all this information together during the pre-license process. And if we see some issues that cause us concern, we may stop the licensing process. And I would say some of our best decisions no one ever hears about because we prevent unsafe vaccines from coming to the market. Typically, though, what the manufacturer and us have looked at the problems early on, and we know what's going to occur. And as we gather our data, we're able to prove safety. But if we see things that are wrong, we'll ask for more data. We may ask for the vaccine to be changed, or like I said, it may stop the licensure process altogether. The other thing we do is if there are some risk associated with the vaccines, we put warning statements on the labeling and restrictions on the products. Warning statements, some common warning statements would be, some animals might have an allergic reaction. There might be a risk of abortion if it's used in a pregnant animal. Restrictions are the type of thing where, like a rabies vaccine is limited to use by a veterinarian, or same way with a brucellosis vaccine, that would be restricted to use by a veterinarian. And some vaccines are restricted by the state or the federal government to only use in very specific situations. And after they've made it through that kind of arduous process of licensure and start selling the vaccine, our safety monitoring continues, right? It does not stop once a product is licensed. And adverse event reporting is a critical part of producing and maintaining safe vaccines. If you as a producer use a vaccine and you're getting adverse events, first you ought to talk to your veterinarian, and two, you ought to call the vaccine manufacturer and let them know what's happening, right? Vaccine manufacturers are responsible for monitoring their products and reporting adverse events to CVB. And CVB has a unit that interprets that data, does its own analysis, does its own investigations. And, adverse events can be, allergic reactions, all sorts of things. And certainly even if you stick yourself with a needle, right, while you're given a vaccine, which does happen occasionally, that should be reported to the vaccine manufacturer, because that's important information and gives us important feedback on the safety. So when reporting adverse events, first always read the label, because on a farm, on a ranch, right, you're going to have vaccines that are regulated by USDA. You're going to have antibiotics that are regulated by FDA. So don't call us about a problem with an antibiotic, because we're not going to be able to help you. So if you read the label, it's going to have the manufacturer's contact information on there, and that would be the first place to start. Certainly, you can also report it directly to us. And that was a really brief overview, because like I said, the following presentations need some time and are going to talk about some important things. I think the key thing, that I'd like you to remember from this is, one, we've had a 100-year mission of making sure vaccines are pure, safe, potent, and effective. And two, read the label, follow the label instruction. And if you do that, the vaccine's going to work well for you. Thank you for your time. I am done with my portion of the lecture.
So as was previously introduced, my name is Dr. Chris Chase from South Dakota State University. And we just heard some really important information from Dr. Thompson about all the things that USDA does, particularly as it looks at what we call fully licensed vaccines. But they've also been looking at some other innovative approaches, and that's what I want to talk about in my portion of the talk. And so what I'm going to talk about is one of the initiatives that USDA has had is in terms of really this issue of having efficacious vaccines that we can have in a timely manner. And one of the things that they have proposed, and they proposed this clear back in 2015, was something called a platform vaccine. And I'm going to explain that in this portion of the talk, because what it allows us to do, it allows us to have a process that we know is indeed very safe and well documented, and at the same time can allow us also the opportunity to get vaccines out in a timely manner, particularly as it relates to your ranch or farm. So I'm going to talk really about two different types of platform vaccines that are out there. But basically, what a platform means is that you begin by taking a target, and I've used BVD and rotavirus, so either one of them, where we actually know that what the important proteins are in terms of causing or of creating an immune response. And we isolate those genes associated with that. And then in the case of what we call this RNA particle, we actually take that gene, put it in what we call a virus helper cell line, and then actually make then a particle that contains the RNA message. So then when it's injected into the calf, that particle can then go ahead, enter into the cell, and then in turn begin to produce viral proteins. And that's shown in this slide. So then once we have the viral proteins, we can induce an immune response. And in USDA, this particular approach is called an RNA particle. And the other type of platform that's been introduced is a platform where the protein, again, the target genes, again, I'm giving you BVD and rotavirus here as examples. So we find, again, the important genes that are on, that we know that are protective. And we take one or two of those genes, we use as many as we need, but we put each gene in individually into a vector system. And then we actually make, in this case, we make the protein. So in this case, the protein is made outside the animal. So in the case of the virus particle, actually it's made in the animal makes the protein itself. In this case, the protein is made outside the animal. It's then introduced in you see an adjuvant on the middle portion of this slide, and then injected into the calf. And I'll get the syringe there. Let me go back to that. There's a syringe for that. And then again, in this case, with the protein in the adjuvant, we induce a vaccine response. And the thing about why we call the platform is when you go all the way back to the beginning here is that you can put different targets. So you could do, you can put BVD in here, but once you have this system put together, you could then put a rotavirus protein in there, another protein for a new virus that could come along. But again, the idea is that you have this basic platform that's been approved by USDA to work with. And so again, you just heard, again, Dr. Thompson talk about the licensing procedure. And when USDA looks at all the things in terms of being pure, safe, potent, and effective or efficacious, that's then called a full USDA licensure. And so again, you take your target protein, you figure out all the age and the route and the disease or the dose, and then put it into this platform. Okay, the system then that you have done all the licensing. I'll show you all the kind of things that they have to go through. And then you also have the opportunity then to actually what we call a swap out and put a gene in from that same particular virus and put it in again, so that you could be able to maybe enhance your vaccine. And you can do that quickly because you've already approved this platform. So let me talk just a little bit more about that. So this what we call this production platform then is, again, as it says, it requires a full USDA licensure. So it has to be what we call non-replicating. They do all these steps that you see in terms of being able to show how to manufacture it, do all the safety and risk assessment that was alluded to by Dr. Thompson that needs to be done, all things in terms of all the levels of safety that he just talked about. And then they do the field safety, what's called field safety studies. And again, they have to show an efficacy. In other words, they have to show how effective the vaccine is. And then once this platform, this series of steps is approved, then within that, because this target gene, again, it could be from that virus, there could be maybe different targets that you want to use, and you move over to the right-hand column, that can be swapped out. And then again, if there's some changes that occur in the field, or we discover there's more things that we need to add, and you can do that with no changes in the manufacturing process. And that really streamlines, as Dr. Thompson alluded to, these things can take a long time from sort of start to finish for full license. So this allows us then to really to be able to sort of speed up the process. And again, you have to have, any full field safety is required if you're going to do it to a different species. And the other thing that we call this idea that you can swap the genes is what we call plug and play in terms of a pathogen. Now, Dr. Williams is going to talk about different types of the fully licensed vaccines here in just a moment, but I want to talk about a little bit more about agents where we don't really have either there are no vaccines or the vaccines particularly are not effective. Again, as much as possible, we always want to use the fully licensed vaccines, because those are products that have been through all the testing that you need, including, importantly, efficacy testing. But with these, sometimes we need to have vaccines that fall, that just we can't get on the market, that aren't on the market, or the ones that are on the market aren't as effective as we need them to be. And so that's when we talk about then having these sort of prescription type vaccines or autogenous vaccines. They require, though, that you have a good relationship already with your veterinarian, with the veterinary client-patient relationship, or the VCPR. They're going to be specific for a premise. And again, the thing is, there can't be any replicating agent. And again, what you might be familiar with is autogenous vaccines, because they've been around for some time, and they, of course, they're inactivated. And then there's the idea, though, that we can take platform vaccines, and now what we go into what we call a prescription vaccine. And so what's the difference with that than just a platform? So again, as you look at autogenous vaccines, and also, autogenous, basically what we look at is purity and safety. There's not anything about potency, and not anything about efficacy. And so so the full licensure gives you the added assurance, particularly about efficacy. But when we do prescription and autogenous vaccines, we don't know specifically what the efficacy would be required. So if you look at then, how can this be used then on a specific premise? That's outlined in this slide. So again, we talked about this full USDA licensure before. The idea that you get this platform license already. So now the system to get the vaccine, or to produce the protein, or to produce the virus particle has been approved. The cells have been documented. All the safety studies have been done with that. And what's the difference then with the prescription platform is that you can take this platform, and now you can put in different antigens from some different pathogens. So again, let's say that it was initially developed for BVD, but on your farm the issue might be rotavirus, that you need a better rotavirus vaccine. Well, with the prescription platform, then what you could do, even though the original vaccine may have been licensed with BVD, you can use this prescription again, but you have to have a valid veterinary client patient relationship. So again, the veterinarian is very important in this process. So again, to use the platform, there has to be already a platform that's been approved, that's already had a fully licensed vaccine. And again, to use a prescription, you've got to have a valid veterinary client-patient relationship. And then again, all the production and all that thing is based on the initial platform, again, which speeds things up. And again, you can use different pathogens with this. And again, it's restricted in terms of the organisms that we have any kind of surveillance or foreign animal disease. So we're talking about viruses and diseases that are endemic in the U.S. And a big advantage, which is the last point here, is about that you can have multiple combinations. So as you see my little cartoon there, I'm showing you the bad bugs of BVD, IBR, PI3, BRSV, and BCV, that you could have combinations within that. So again, the advantage then by using this platform is that you have a consistent manufacturing process. It's biologically relevant because, again, if there's a change in strain or strain that's not specific to your farm or ranch, that's an opportunity. You can look at sort of new and emerging disease options where we don't have, let's say, any kind of a licensed vaccine. They can be very customer specific. And the thing is, one of the things that we have problems with some pathogens, it's just hard to grow them. So all we need here is we just need to be able to get a hold of the target gene that we're after, and we have a lot of the tools of molecular biology to help us with that. So it makes it much simpler than to actually use this kind of a platform for these types of issues. This is just showing you the idea then that if we look at a whole cell versus a subunit. So whole cell would typically be if we had a autogenous vaccine where we grew the virus up and then we inactivate it, so you end up with the whole organism, and the whole organism then is put in, whereas prescription vaccines only going to have the target of the part that we know is the most important. And in this case, they're showing you just the one protein called hemagglutinin. So they've taken the hemagglutinin gene, put it into the platform, and just been able to produce that as the antigen. Just the last couple slides here then. So what's some advantages compared to commercial versus the prescription? So the big thing, and again, Dr. Thompson really went through a lot of the important things that we understand, particularly is safety. But we know the commercial vaccine, again, it's the full licensure has occurred, so we know it's pure, we know it's safe, but most importantly, like that last point, we know that it has proven efficacy. But the other issue that we know is that these licenses can take some time. Compare that then to the prescription, then we know again that we can take just the gene that we're interested in, that targeted sequence, and in those viruses that are hard to grow, we can just, again, all we need is just to have that thing, that particular protein. We use an adjuvant with this, and again, we need to have that veterinary client relationship, and again, we can target it, and the development cycle then is much shorter than what it would be with a fully licensed product. But again, you look at that last point, they're going to be safe. The efficacy is going to be unproven, where the initial platform for that particular protein was shown with efficacy because it's fully licensed. These will again, you're going to see how it works on the farm to see how efficacious that it is. And then compare that then to the other type of sort of prescription type of vaccine we can have, and that's autogenous. And again, the big thing here is we've got just a targeted gene sequence. We can, again, in both cases, you're going to have a veterinary client relationship. You get the target that you're after. The safety is going to be well proven with this. And again, with both of these, you're going to have efficacy is relatively unproven. With autogenous, the other issue there is in terms that you're killing the entire organism, and sometimes there's things in that organism that are not necessarily what you want to have. And if it's a organism that's hard to grow, of course, that's another issue as well. And again, and we look at, again, with vaccine development, and I think, again, I want to applaud USDA and the CVB for being very, very forward thinking about the fact that, having this type of opportunity really provides, better opportunities to have new and better vaccines. And so the points here about, there could be diversity. So again, maybe that vaccine, that the fully licensed one, doesn't work quite at the level you need on your farm. So it allows us to do some changes there. There's differences within the countries or continents. We can get on that actually much quicker because all we need is just to have that genetic sequence. And again, that last point about growing these organisms that are hard to grow, this really gives us a big advantage with that. And again, I just would just close with just saying that, I mean, again, USDA has been very proactive about this new and different process in terms of registering vaccines, which really has allowed veterinarians and the U.S. biological companies to really be able to help us out in a very timely manner by, again, having the pathogens isolate specific, for a farm. The approval process has been streamlined, and you can go after the things that are most important. And with that, I would like to now turn it over to Dr. Willems to wrap it up for us with what I call the big show. Amelia.
All right, thanks very much, Dr. Chase, and thanks everyone for being here tonight. I really appreciate being invited by the NCBA leadership to participate in this webinar, and I hope that you will find it useful. I'm going to try to add a little bit of specific detail to what Dr. Thompson and Dr. Chase have already told you. And let me just get my presentation set up here. And so the specific detail I want to talk to you about is related to some certain specific kinds of vaccines. But before we talk about specific kinds of vaccines, I thought it might be helpful to just kind of review what exactly are vaccines and how exactly do they work. We'll be talking a little bit about RNA vaccines. So before we do that, I want to talk a little bit about what exactly is RNA and how does it work. And then we'll finish with talking a bit about how RNA vaccines work. So we're talking a lot about vaccines tonight. And I think it's just important to kind of remind ourselves that the point of vaccination is to try to help the cow's immune system. And the cow's immune system has to protect her on a variety of fronts. It has to protect her gastrointestinal tract. It has to protect her respiratory tract, her mammary gland, and her urogenital tract. It has to protect this cow on all these fronts, and as well, her whole body surface. And all this is done by things that you cannot even see with the naked eye. The immune system has to protect the cow against infection with a wide variety of different kinds of bacteria and viruses and parasites and fungi. The immune system has to try to keep those organisms out of the cow. But if they happen to get into the cow, the immune system has to clear them without harming the cow or her unborn fetus, which she is often carrying. So this is really an amazing a feat that the immune system often carries out without us even thinking much about it. So the immune system protects the cow or any individual as we all have an immune system to protect us against infection on all fronts. And it does this by recognizing when something that is not self, that can cause danger, enters the body. The immune system activates different kinds of cells that have different jobs to get rid of the not self thing, usually an infectious agent is what we're talking about, to clean up any damage caused in the fight against the infectious agent, to help the immune system remember this infectious agent and prepare to keep it out in the future without causing too much damage in future fights. So these are all the things that the immune system is doing. This all happens when an infection occurs. But when an infection occurs, while it may activate a very useful immune response that can protect the cow or any other individual in the future, at the time of infection, it can make the cow very sick and maybe even kill her. A vaccine can activate all the same effects as an infection without making the cow sick or killing her. And so that's why we vaccinate, to try to get the benefits of immune memory without the harm of a natural infection. Now, there are many different types of vaccines, and Dr. Thompson and Dr. Chase have talked about some of these. And so just to kind of recap, we have killed or inactivated vaccines, and that's what we call them when they contain a virus. If they contain a bacteria, we sometimes call them bacterins. We've also got modified live vaccines, and we've got toxoids. And I'm sure many of you on this call have administered killed or modified live vaccines to your cows, probably even toxoids. The Clostridial vaccines often contain toxoids. We have many killed, modified live, or toxoid vaccines licensed in the US for use in cattle and other animals. Now, there's some other types of vaccines, for example, subunit vaccines, gene-deleted vaccines, chimeric vaccines, or vector vaccines. And these are licensed in the US for use in other animals, but we don't currently have any license for use in cattle. DNA vaccines are another type of vaccine, and these are licensed in the US for use in poultry and in dogs to prevent cancer or melanoma. And then we have a small number of RNA vaccines that are currently licensed in the US as prescription platform products for use in swine. And that's what Dr. Chase just talked to you about, those prescription platform products. One of the currently licensed prescription platforms is an RNA vaccine. So what is RNA? Well, RNA is in every living thing on Earth. The cells that make up every living thing on Earth make RNA. Now, you probably understand that living things are made up of cells. Some are made up of only a few cells. Many of the things that we all work with, like this calf, are made up of millions and billions of cells. And these individual cells are too small to see with the naked eye, but they are incredibly busy. And so this is one cell magnified. And what it's showing you is that every cell has in its nucleus DNA, and DNA in the nucleus of the cell is the blueprint for all the proteins the cell can make. Proteins are essentially the tools that the cell uses to do its job. And if it happens to be an immune cell, the immune cell uses proteins to protect the cow against infection or to clear infection if it occurs. Now, DNA is copied to a template called RNA, and it's actually the RNA template that the proteins are copied from. After the proteins are made, the RNA is broken down by enzymes inside the cell. All the bacteria and all the parasites that can make cattle sick contain RNA. So anytime a cow is infected with any kind of bacteria or any kind of parasite, that cow is going to get exposed to new RNA that's in the bacteria or the parasite. Some viruses also contain RNA. For example, bovine viral diarrhea virus, or BVD, bovine respiratory syncytial virus, or BRSV, rotavirus, and coronavirus. These are all RNA viruses. Now, there are some viruses that don't contain RNA, but they contain DNA, and IBR virus is one of the DNA viruses. But when a DNA virus infects a cell, it causes the cells to make RNA. So here's a little kind of a cartoon to show you how this all happens. Over in the top left of this image, you can see a little cartoon of a virus. In this example, it's an RNA virus like BVD, BRSV, or coronavirus. When the virus infects a cell, RNA starts to be made. That virus starts to copy RNA to make proteins that will then become new virus. And that new virus emerges from the infected cell and goes on to infect other cells and possibly to spread to other cattle. When we give a modified live virus vaccine, this same sequence of events occurs. The modified live virus in the vaccine starts growing and making RNA, proteins, and new virus. And the immune response to this vaccine virus protects the cow from future infections. And so here's another little cartoon kind of demonstrating this. Now I've got on the left some virus, some cartoon virus, but the vaccine vial and the syringe indicates that this is now modified live virus in a vaccine. So when we give a cow a modified live virus vaccine, that virus infects the cells. RNA from that vaccine virus is made. Proteins from the vaccine virus are made and turned into new virus. And the immune system sends cells in to break down those viruses into proteins and RNA, and that leads to activation of a protective immune response. Now, so modified live vaccines have been fantastic to help prevent disease due to a lot of different infectious agents, but they can have problems. Sometimes the modified live vaccine can be too much like infection, and the cow might develop a fever, go off feed, or have other problems. So why not use killed vaccines? They're not going to replicate like a live virus. Well, sometimes killed vaccines don't induce effective immunity. So you might ask, was there something in between a modified live vaccine and a killed vaccine? RNA vaccines are an option between a modified live and a killed, because RNA in the vaccine induces protein production like a modified live vaccine, and so the immune system responds like it's a real infection. But the RNA vaccine doesn't code for the whole virus, so you don't have whole virus made that might actually cause problems. So here in this little cartoon, we've got up in the top left some RNA molecules that are in an RNA vaccine. And this is going to be RNA that codes for one of the proteins on a virus that we know is going to activate immunity. And so when we give an RNA vaccine, that RNA goes into the cell. Proteins are made from that RNA, in this case, the protective proteins for a virus. And the immune cells come in to break down those proteins and ultimately break down the RNA as well to activate the immune response. The RNA and protein later on is completely broken down by naturally occurring enzymes in the host body. So there's some real pros to RNA vaccines. They provide benefits of modified live vaccines without the risks of modified live virus whole virus exposure. And RNA vaccines can be safer in terms of being less reactive, but also more effective than killed vaccines. So if you know the proteins that are important for immunity to a virus or even a bacteria, RNA vaccines can be made very quickly. And that's what Chris was talking about earlier, that the use of RNA vaccines could enable very rapid development of effective vaccines. Now, like everything in life, where there are pros, there are also cons. Some of the cons of RNA vaccines are that they may require carriers made of lipids and proteins to ensure effective immunity. These are similar to adjuvants that are included in currently available vaccines. And figuring out the right carrier can take time and money. Another con at this time is that RNA vaccines are more expensive to manufacture than conventional modified live or killed vaccines. But prices of a lot of things go down the more we make them, and so we may find that RNA vaccines are cost-effective for use in livestock in the near future. So for tonight's webinar, a few take-home points. Vaccines approved by the USDA are pure, safe, potent, and effective. It's been the case for the last 100 years. RNA is present in all living things, and RNA enters the body during natural infections or after vaccination with many types of modified live vaccines. And during the immune response to infection or vaccination, RNA is broken down by naturally occurring enzymes in the body. And the last important point, cattle producers need innovative approaches to provide effective vaccines that can be available in a short time frame. Thanks very much to all of you for your attention, and I hope we can answer any questions you might have.
Excellent. Thank you all to all of our speakers for everything that you just presented. I think for those of you who don't know me, I'm Julia Herman. I'm the beef cattle specialist veterinarian here at NCBA. I feel very fortunate to be able to work with professionals in the field like this and have them come bring this new information to you. And we have a lot of questions, and I'm going to start off with one of the basic ones that, or not the basic ones, but it's more of a definition. What does RNA and DNA stand for? And I'm going to have Dr. Willems discuss that since she just got done.
All right, so thanks, Dr. Herman. So RNA stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid.
Those are proteins that are found in all cells, correct?
Well, technically they're nucleic acids, so a different type of a molecule, but they are indeed found in nearly all living cells. I say nearly all because, for example, mature red blood cells that don't have a nucleus don't have any DNA, but they do have RNA in them. So almost all living cells are going to have DNA and RNA.
Thank you for that clarification. I'm going to step into some vaccine handling. I think a lot of the listeners out there likely have gone through the beef quality assurance program. Has a lot of points that our speakers talked about today. Having that veterinarian client-patient relationship, reading the label on all of our animal health products is really important. I'm going to have Dr. Chase answer this one. What steps should be taken to eliminate an animal reacting to a vaccine, which results in a hard knot on their neck?
Well, thanks for the question, Julia. I think one of the things that you want to make sure, because obviously, hopefully, in the formulation when it was developed in terms of being safe, one of the things that was looked at is, and I can tell you that because I've been involved in these these safety studies, is to observe the inoculation site in terms of vaccination site. So I think one of the things that I've seen where we've had issues certainly is needle usage, and in terms of reusing needles, having needles that they may have either get a burr on them. Needles that end up, becoming, contaminated. That's been a big issue. We know that certainly as you look at, when cattle and weather, when we certainly have more wet weather, we're gonna have more issues in terms of just, as we make that inoculation, increasing the opportunity that we can introduce, other materials that are going to result in that, in a knot and those kind of things. But I think I would really begin with needle usage. To me, is probably, as I look at, in terms of burrs, or just, not changing frequently enough, and having, if we have wet hides and some other issues in terms of contamination, that can certainly enhance that. So if we can avoid that, that would really help in terms of decreasing that kind of reaction that we get that results in a knot.
Oh, that's great advice. And one of our other questions goes to vaccine handling too. They ask, once a vaccine is taken out of the fridge, how long is it good for when working cattle?
I'll go ahead and I'll start with that, and then let anybody else chime in. So it depends. So if it's a modified live vaccine and we reconstitute that vaccine, it has a relatively short shelf life in terms of we're talking about keeping it protected from sunlight, keeping it cool. And typically, we're within a few hours after that. Now, with a inactivated vaccine or Bactrins that are frequently in a multi-use container, there again, it's important that you keep it refrigerated or cool, and again, out of direct sunlight. And of course, they will have a little bit different labeling in terms of how long that what the expiration date is. But the problem, of course, that we have with those is just the repeated reintroduction in needles, can causes an issue. But the modified live, in particular, ones that we need to be cognizant of. I mean, if you reconstitute and then put it back in the refrigerator to use it tomorrow, that's not a good practice to do, because the efficacy of that vaccine is going to diminish relatively quickly. And I don't know, Amelia, if you have any other comments you want to add to that.
No, I think you covered it really well, Chris.
Excellent. The next question, we're going to pop over into bacteria. I know we've talked a lot about viruses. We had a question about they have a lot of wild elk around them, and those elk have been known to carry different bacterial diseases like Johne's. We know that some elk herds are managed a lot like cattle in some parts of the country. Could cattle vaccines be effectively used on elk herds, if that's an option?
What the USDA standpoint is, the label of the vaccine says what it's used for, but the regulations allow for veterinarians under, using their professional ability to make good decisions to use vaccines as they choose to use them. In this case, potentially as an off-labeled use. And certainly in some of our minor species, right, that occurs commonly. But when that is done, right, it's the individual veterinarian taking on the responsibility, knowing their vaccines being used in a species that it may or may not have been tested on. See, it really falls solely on their evaluation of the herd, the situation, and the vaccine.
Yeah, I think having that veterinarian in there, to decide, what the diagnosis is, what to treat with, and the proper tools to treat that with, that's a very good point. Thank you, Dr. Thompson. I'm going to ask Amelia this next one. How do you know that you have the correct target gene or antigen that will induce a protective immune response? And I think this was towards the platform, but I think it could be incorporated into the other vaccine types that you talked about.
Yeah, that's a great question. So it's going to take some research to answer that question. And a lot of times that starts out in a basic lab, like in a university or possibly in a government laboratory. People that are studying the different pathogens, they study the different components of the pathogens and then do experimental vaccine trials to determine which components of the pathogen are the best targets for an effective immune response. But we don't know that for every pathogen. So for some organisms, it's pretty clear. For others, we may not have all the information we need.
Always need new research. I think that's an important lesson. I think just from this, one of the important lessons that I'm taking from this webinar, that relationship we have between USDA researchers like you and the producer and veterinarians out in practice. I think that's a really great relationship that we can all learn from each other and really move forward, taking care of our cattle as well as we can with the best science that we have. I have one question. I think I'll ask Dr. Chase this one. Other than immune system cells, what other cells or organs might RNA platform particles enter, and what effects might that create other than the desired immune response? So potential side effects.
Well, I mean, that's a really good question. We know that. I mean, typically when we give the types of adjuvants that we use and the other things that we put in like with the virus particle vaccine, that they're going to they're probably going to go into other cells as well. I mean, if I'm giving it intramuscular, I can just about guarantee you that it will go into some of the muscle cells. But the thing that Amelia spoke to is the issue that whether it's a protein or whether it's RNA, that the cells have a system to degrade that. So in the cells that are not immune cells, and typically we think of what we call antigen presenting cells, or cells whose job it is to pick up that antigen and present it to the rest of the immune system. Those other cells are, I mean, they may express the protein a little bit, but the reality is that, and that may itself be picked up by immune cells that are in surveillance, but its longevity in those cells is going to be really short, because again, all to the cell, RNA is not. And why when we think about RNA, most of the time we often use the term messenger RNA, and the reason we use that is because in the cell, messenger RNA has incredibly short half-life, because when it makes a protein, it's degraded, and so even in some in non-target cells, some of them may produce protein, which then can be picked up by the immune system, but in general, there's just not going to be much. It's going to be degraded, and really won't have any long-term effect on those cells.
That was a nice explanation. I have one question that I'm just going to answer I'm going to ask to the group. So we know that there were a lot of mRNA vaccines were used for COVID vaccines in humans. This is similar, but not exactly the same technology. But if we do use these platform vaccines eventually in cattle, we know that they're not being used right now in cattle, or other RNA vaccines in cattle potentially. How do we make sure that the meat from those animals is safe to eat?
So I'll go ahead and start. I mean, there's lots of ways to answer it, and I think the first thing to remember, as the other presenters have said, is whether it be the immune system is designed to recognize and eliminate foreign RNA, and it does it on a daily basis, just as we exist, because we're always being exposed to pathogens. But the way USDA is going to look at it is we're going to do that overall risk assessment, and when we do that risk assessment, right, part of that is the public health assessment of, is there a probability of human exposure, whether it be through an inadvertent needle stick or ingestion of meat, and then what would be the risk of that, or possible outcome of an inadvertent exposure. And so all of our safety tests that we talked about, including the injection site studies, it's really looking at the entire big picture, because certainly USDA knows that having a wholesome meat product is critical for a vaccine.
Julia, I'll go ahead and add that the situation is really very similar to the situation when we give a modified live vaccine nowadays. So when we give a modified live vaccine nowadays, that's got virus that has RNA in it in most cases. Or if it's a DNA virus like IBR, it'll get into the cell and start making RNA. But if we give a BVD or a rotavirus or a BRSV vaccine that's modified live, we're creating RNA inside the cow. And so one of the things the USDA does to just make absolutely sure that there's no problem with the meat is to have a withdrawal time for the vaccines. And so vaccines have withdrawal times, usually between 21 and sometimes as much as 60 days. And that is a way to give the body of the cow plenty of time to break down that RNA like we've been talking about.
And I would just add to that. I mean, so Amelia just said in terms of what we're using right now in terms of, and I would say one of the things that when I have the opportunity to go to many different countries in the world, and we're one of the few countries that actually, when it comes to vaccines, that actually we put that that withdrawal on there, and that's just to put it in really an added measure of safety. I think from a risk assessment standpoint, a lot of other countries don't have that at all. And I would just also say that, I mean, certainly in terms of ingestion and anything else, where we're taking in RNA all the time in our our digestive tract and everything else. One of the other things it does is it breaks down all that, and as cooking and other things do. But I think just in general, we have been using in the levels of RNA that you get from a modified live vaccine are going to be at levels, orders of magnitude higher than anything that we would do with many of these platform vaccines. So to me, it's just I think we have really done a good job of really trying to minimize the risk, and at the same time give ourselves plenty of margins of safety.
Thank you very much. That was a wonderful explanation. I think withdrawal times comes down to reading that label. So Dr. Thompson mentioned that earlier. I tend to repeat that to myself a lot. So hopefully other people are taking that lesson also. I know we're just a couple minutes over, but the audience has really been giving us a lot of great questions. So we're going to go just a few more minutes. Are there any RNA vaccines given orally on the market?
So I can, yeah.
Go ahead, Chris. Yeah,
yes,
go ahead.
Yes. So the answer is certainly there's a rotocoronavirus that is given to young calves that's given orally, and of course there's a number of them that are given nasally, and those are modified live. But yes, certainly, because you can certainly imagine that if I give something nasally, that a lot of that can end up going orally. But there's also, like I said, there's a rotocoronavirus vaccine in calves that's administered orally. So.
Yeah, I was just going to say that I can't think of any true RNA vaccines that are given orally at this time, but certainly the rotacorona vaccines that are given orally are a good example of a virus with RNA that is going to then replicate and cause the production of more RNA in the intestinal tract.
Excellent. This one is a really great question. I think it focuses on BRSV, or bovine respiratory syncytial virus, but I think this can apply to a lot of viruses that we have out there. The question is, we know that the historic vaccine lines are not keeping pace with the disease. So as we lose availability of new antibiotics being produced, how can we have more effective viral and bacterial prevention when we don't have those tools to treat the disease?
Chris, you want to take that?
I'll start and I'll let you come in. So it really, again, the point about having, and again, when you have a lot of fully licensed vaccines, I mean, again, they're always our first line of defense because we know the efficacy of those. But we know, as the question sort of points out, it's a good question, that some of these, the thing, unfortunately, when it comes to viruses, and RNA viruses in particular, is that they have a high propensity to change. And so, what was effective before may not be as effective. And so being able to sort of continually streamline and update a vaccine, and that's really where the platforms have a lot of value, because their ability to be able to update is really rapid. Once you have the platform, you're just, as I mentioned before, the plug and play. So I mean, to me, that's why vaccines become even more important tool to us, because as we lose the use of antimicrobials, we need to, what else can we do to enhance that animal's normal immune response, so that animal's not as dependent on antimicrobials? And Amelia, add anything else that you'd like to. So.
Well, I just wanted to say the question touched on the idea that viruses may be changing and vaccines may not be keeping up with those changes. And that certainly is an important issue with some viruses. And I've certainly talked to people that think that could have been a problem in sort of individual examples where cattle vaccinated for BRSV went on and then got BRSV. But I did want to say that people have looked at sequences of our BRSV over the years, and it doesn't change as much as other viruses, like, for example, BVD, which we know changes quite a bit. But we need research to look at this. And Julia said earlier, you know, we always need more research. And some people say, well, that's all you researchers want is more research. But it would be nice to know more about the sequences of BRSV. And I just wanted to say that for those of you who might ever talk to people in Congress, let them know that we really need that USDA research funding, because USDA research funding is where we figure out what kind of antigens we need to have in vaccines and whether or not viruses are changing in ways that require new developments in technology. So.
I'm going to do 2 more questions, and then I think we're going to have to wrap it up. But the first one, I think it's definitely a question I've talked with a number of people about, but can enteric coronavirus be protective against respiratory coronavirus infection? And I'm going to say in cattle, since that's what we're talking about.
Well, I'll start with this one. That's you know, $1 million question. I mean, the thing that makes coronaviruses unique, especially in livestock, and this is true in cattle and swine, is that they're what we call them pneumoenteric, I mean, which they can affect both the respiratory system and the GI system. So we certainly know that is a you know, in the pig world, we know that there actually was a virus called TGE that mutated and became a respiratory virus and now is protective. I think it really kind of depends on the situation, and then this kind of gets you back to the coronavirus that you're dealing with. But I mean, I think because they are pneumoenteric, that you can certainly see some cross protection, as we're aware of, though the vaccine is only labeled for enteric use. But again, the virus has this trophism to both be in the gut and the respiratory tract. So I think, you know, it's an area, if we talk about more research, it's another one that we need more research. And Amelia, I'll let you take a shot at it.
Yeah, I can't really add too much. I guess I would just say that I have heard of anecdotal situations where people have tried the enteric coronavirus vaccine to help decrease respiratory disease, and they feel like it's been helpful. For some of the vaccines, that would be an extra label use, and so you'd need to talk to your vet about doing that. So there is some evidence of that.
Thank you for that explanation. I'm going to combine 2 questions into this last question. It'll be a group effort. So research shows that vaccination with multiple different viruses and bacteria at the same time can sometimes induce the wrong kind of immune response that could potentially exacerbate a disease. How can we reduce the risks generated, or how can we reduce the risk of generating inappropriate immune responses that may be harmful to the animal? The other part to this question is another person asked, are you concerned with antigen overload by adding in so many antigens onto one platform? So I think we can answer it towards the platform vaccines, but also just in general, can you give too many vaccines at one, or too many antigens at one time to an animal?
All right, well, I'll start. So one of the advantages when you talk about platforms is the idea that, you know, there was a good question asked before in terms of, you know, how do you decide what the right target is, and you know, if we can, you know, if we get these down to, you know, 1 or 2 proteins for that pathogen, you know, in terms of the amount of antigen is actually considerably less than when you give the whole organisms. So that's certainly, and the thing that we know, and again, this is really important when you look at a combination vaccine that is labeled as a combination vaccine, all of what we call the interference work has already been done with that in terms of showing that you know, there's no interference, at least with that vaccine. But of course, the problem becomes then, because we know that every time we vaccinate an animal, that's a stressor. And so we want to try to minimize that by handling them less and then giving them maybe 2 or 3 different products at a time. And that can be problematic. I mean, that's where we get into, particularly with gram-negative bacteria, especially several of them, that certainly can cause some other additional, because we know, particularly gram-negative bacteria, that endotoxin is an immunostimulant, and too much of that is you know, I refer to that as sort of too much of a good thing. So it's you know, the products that are labeled and that are combination have already been shown in terms of being efficacy and interference, but you know, that becomes problematic as we use different products at the same time. And that, you know, one thing that I would just say, and then I'll stop, is that one of the things that you can do that will, I think, has some utility, and that is literally breaking it up. So give on one side of the neck and then give the other one on the other side of the neck. There's certainly some utility to that, because all the action for these vaccines basically occurs in a draining lymph node, but that still doesn't, you know, overcome, again, that there's some things where especially it's a lot of gram negatives, and I'll stop there.
Yeah, I was going to add something there, but Chris did such a good job of explaining our processes, where in the sense of, you know, if you have a combination vaccine that's all in one package, CVB will have done the testing to prove that there isn't a loss of efficacy. But yeah, when you're at the side of the chute and you're drawn from 3 different vials, right, CVB hasn't done that testing, because you know, there's almost an infinite number of combinations that could be given to an animal. So, you know, the labeling for the vial states what it, you know, what the data supports. So I'll leave it at that.
And I'll just say one last thing quickly, because there has not been and probably never will be testing of all the different combinations of different products you could give to a cow at the same time. I think it's best not to give any more vaccines than really seem to be necessary. I have great confidence in the licensed vaccines we have in the United States, but every cow doesn't need every one of them. And so I would say work with your veterinarian to figure out if there's really a good reason to give a certain vaccine. Don't throw another vaccine into the mix just in case, because it might end up being less helpful than you might imagine. So I would say try to work with your vet to kind of know what sort of disease risk you have on your ranch or farm, and really just focus on vaccines for the agents that are really likely to be a problem for your operation.
I think that is an excellent point to end this really, really educational webinar on. I want to thank all of our panelists for joining us today, and the NCBA producer education team is always looking for other topics that you, the producer, are interested in. And the topic of this webinar came directly from our members. So if you have any other questions or comments, you can contact NCBA. But thank you all for joining us, and I hope you have a great week.